The next anti-aging aid is xenohormesis and microRNA manipulation. Xenohormesis and microRNAs represent cross-kingdom communication pathways between plants and animals that we may be able to use to our advantage. Xenohormesis. Hormesis can be thought of as the "that which doesn't kill you makes you stronger" principle. Physical activity is the classic example. You put stress on your muscles and heart and are all the healthier for it, provided there's sufficient recovery time. Mild stresses like exercise can trigger a protective response that leads to strengthened defenses in the long run. In the 16th century, the Swiss physician Paracelsus, the so-called father of toxicology, coined the Latin phrase sola dosis facit venenum, which is translated as, quote, "only the dose makes the poison," unquote. This aphorism is typically summoned to explicate how some of the most helpful or harmless substances, like water, can be toxic at high enough concentrations, and conversely how even some of our most poisonous substances, like cyanide, can be harmless at sufficiently infinitesimal doses. The field of toxicology adopted this threshold dose-response model, where at low enough concentrations there may be no effect, but above that the hazard is proportional to the dose. Hormesis adds a new wrinkle, forcing toxicology to challenge this assumption. Hormesis takes the notion too much of a good thing can be bad and turns it on its head by suggesting that sometimes a little of a bad thing can be good. Hormesis comes from the Greek term hormein, meaning to excite. Rather than a linear model in which there are small effects at small doses and the same but larger effects at larger doses, Hormesis describes a biphasic response characterized by one effect at a small dose and the opposite effect at a higher dose. For example, herbicides kill plants, but in tiny doses they can actually boost plant growth, presumably by stressing the plant into rallying its resources to successfully fight back. What began as a biological curiosity used in a misguided 19th century attempt to vindicate homeopathy is now undergoing a resurgence in interest. In the 1980s, as few as a single study a year was published on hormesis in the scientific literature. Now, on average, more than one paper is published every day. This is in large part due to interest in the role of hormesis to combat aging. Bask in the glow? Hormesis was first shown to extend life more than a century ago when low doses of radiation were found to increase the lifespan of beetles by presumably ramping up DNA repair. That which didn't kill them made them stronger. I run through the wild ride of a story in c.nf/radiation, including studies suggesting longer lives among atomic bomb survivors, and experiments done more than a mile down into the Earth to counter the cosmic rays bombarding us every second. Nothing in life is to be feared. Marie Curie, who won the Nobel Prize for pioneering work in radioactivity, is quoted as saying, "It is only to be understood." Of course, this is coming from a woman who died of bone marrow failure from radiation exposure, such that her remains had to be interred in a lead-lined coffin. Watch the video, but the bottom line is we don't know enough about low-level radiation to exploit any hormetic effects without potentially being exposed to unacceptable risks. There are, however, salutary ways to harness hormesis for health and longevity. No pain, no gain. We all know that exercise is ultimately good for us, but nonetheless it places inseparable stress on the body. Ultramarathon runners generate so many free radicals during a race that they can damage the DNA of a significant percentage of their cells. But within 1 week, they don't just go back to the baseline level of DNA damage. They have significantly less damage, presumably because they've revved up their antioxidant defenses. So exercise-induced oxidative damage may ultimately be beneficial. In other words, classic hormesis, where low levels of damage can upregulate protective mechanisms and ultimately leave you better off. For those interested in how not to undermine the benefits of athletic recovery, check out c.nf/exercise hormesis. That which doesn't kill plants may make us stronger. Hormesis may be why dietary restriction can lead to lifespan extension. The mild stress placed upon the body by not eating enough may activate a wide variety of protective pathways, ramping up anti-inflammatory and antioxidant defenses. Your body is preparing itself for the coming famine it thinks is about to occur. In the caloric restriction chapter, I'll explore ways to exploit the benefits of dietary restriction to extend life and prevent disease. But chronically restricting food intake is not a realistic health strategy for many. Given the powerful evolutionary drive to eat, it's hard for most people to cut food intake by even 10 to 20%. A more feasible alternative may be to activate dietary restriction-induced stress response pathways by other means. One such possibility is xenohormesis, derived from the Greek xenos, meaning stranger, foreigner, or other. Xenohormesis refers to the bestowal of stress resistance from stressed plants to the animals who eat them. In other words, instead of exposing ourselves to the stressor to trigger our body's defenses and shore up protection against future stressors, why not let plants take the hit? Couch potatoes don't have anything on real potatoes. Plants live the ultimate sedentary lifestyle. Because they can't move, they've had to evolve a whole other way to respond to threats, and they do so biochemically. They manufacture from scratch a dizzying array of compounds to deal with whatever's coming their way. If, for example, we get too hot, we can move them to the shade, but if plants get too hot, they're stuck. They are the shade. Plants have had nearly 1 billion years to create a whole chemistry set of protective substances, some of which can play a similar role in us. After all, where do most vitamins come from? Plants make them for their own needs, and we hijack them for broadly analogous cellular roles in our own body. There's also a shared set of so-called vitagenes conserved through evolution to encode an array of repair and maintenance processes, such as heat shock proteins that confer fitness and survival benefits. Nature programs marvel how closely we're related to chimps, but we share about 1/5 of our genes with a banana, even though it's been more than 1 billion years since we've had a shared ancestor, before the human and the banana split. Nature didn't reinvent the wheel for critical cellular processes, such as basic metabolism and preserving DNA integrity. Plants and animals even share some of the same stressors. We get attacked by bacteria, and so do plants and fungi. When bacteria muscle in on a particular fungus, it creates a molecule called penicillin, provided free for us, and when a fungus muscles in on a particular bacterium, it produces rapamycin as an antifungal to slow its growth, inhibiting the target of rapamycin, TOR pathway, conserved in fungi, plants, and animals, including us. Remember, that's the same engine of aging enzyme pathway that can be tweaked to extend lifespan. When plants get infected, they produce the compound in aspirin, which can come in handy when we get infected ourselves. Plants heal wounds, and so do we, using similar signaling systems. Plants have DNA they need to protect from free radical damage, so they cook up complex antioxidants that we can use for ourselves instead of reinventing the wheel. In a sense, the CRISPRs in our fridges are like nature's medicine cabinet. We can just let the plants get stressed because, incredibly, the stress response molecules in plants may activate the same protective responses in us. The majority of known health benefits of edible plants may be attributable to the pharmacologically active substances of plants' sophisticated stress responses, off which we can then piggyback. For example, I've often mentioned polyphenols, a class of phytonutrients for which there's a huge medical literature on health-promoting effects. Plants produce polyphenols to protect themselves, and we may be able to expropriate and commandeer them for our own similar purpose. Xenohormesis explains how environmentally stressed plants produce bioactive substances that can confer survival benefits for those of us who consume them. Drought-stressed strawberries, for example, have more antioxidants and other phytonutrients. Have you ever eaten a wild strawberry? The taste is incomparable to the flat flavor facade of the cultivated kind. The healthiest grapes often grow in relatively dry, sun-exposed, infertile soil. Studies show that commonly consumed fruits and vegetables can be nutritionally enhanced by light, water, nutrient deficits, cold stress, or being nibbled on by bugs. That may help explain why the levels of phytonutrients are estimated to be 10 to 50% higher in organic vegetables compared to conventionally grown ones. Organic grape juice, for instance, contains more polyphenols and resveratrol than conventional grape juice. Similarly, soups prepared from organically grown vegetables contain levels of salicylic acid that are nearly 6 times higher than soups prepared from non-organic ingredients. If you starve plants, they do the same thing mammals do, activate preservation pathways. So let the plants face the adversity to create the molecules that trigger cell stress resistance, alter metabolism, and improve disease resistance. Then we can just capture them for the same uses in our own body. The fact that many phytonutrients act as quote-unquote dietary restriction mimetic, in that they mimic the physiological effects of dietary restriction, may be no coincidence. The plants produce these compounds to save their own green butts from scarcity. So instead of having to starve, thanks to xenohormesis, we may be able to let plants bear the brunt and enable us to harbor their hardships as a means to promote our own health. Plant pokes. The flip side of xenohormesis is that plant compounds themselves can act as a source of hormetic stress that ends up bolstering us. If you remember from the oxidation chapter, green tea's rallying of antioxidant and DNA repair defenses appear to be a consequence of its mild pro-oxidant qualities. It ends up doing a lot of good by being a little bad. The constant small jabs with every sip rev up our defenses to better protect us when a more serious insult comes along. It's like minor irritations that build up calluses on our hands to fortify their resilience. The end result? Interventional studies on rodents show that green tea extends their lifespan, and observational studies on human populations show that tea drinkers may average lives that are years longer. Remember the broccoli story from the same chapter? How the cruciferous compound sulforaphane is the most potent natural inducer of NRF2, the guardian of health span and gatekeeper of species longevity? Our body wouldn't ramp up the detoxification enzymes in our liver every time we ate broccoli if it didn't consider broccoli to be a threat at some level. It's like how applying the red pepper compound capsaicin on our skin can trigger heat receptors and fool our body into sweating and flushing its way to actually bring down our internal body temperature. Our body seems to picture each floret of broccoli as a miniature medieval mace and responds by battening down the hatches. We can then reap the rewards of this veggie vigilance and enjoy a longer life as a result. It's no surprise that our body is finely tuned to react defensively to so many compounds in plants. After all, plants don't want to be eaten. Sulforaphane is thought to be created in plants to dissuade nibblers with its bitterness. Allicin, the garlicky compound in garlic, is presumably made for the same purpose. At petri dish concentrations exceeding that which could be achieved by even garlic lover levels of consumption, certain garlic compounds can be toxic to mammalian cells obtained from human foreskin, so no applying raw, crushed garlic to your skin. But at culinary levels, our body evolved to handle subtoxic dosing in pasta sauce and induce the adaptive stress responses thought responsible for garlic's health benefits. Or whether some of our healthiest plants are actually mildly toxic, or our body merely treats them as such, the end result is the same: health through hormesis. Power plants. There are thousands of phytonutrients that will never make it onto the side of a cereal box, yet may play a role in reducing the risk of chronic diseases, and those are just the ones we know about. The terms phytonutrient and phytochemical refer to natural compounds found in plants that can affect our health. Phyto comes from the Greek phyton, meaning plant. They're not considered quote-unquote essential nutrients like vitamins, as we can technically survive without them. Instead, they've been called quote-unquote lifespan essential, meaning they're necessary for the longest possible life. In this way, they're like dietary fiber, critical for optimal health and longevity, but not technically essential, as coma patients can survive for years on an intravenous mixture of sugar water, electrolytes, amino acids, vitamins, and a few essential fats and trace minerals. How many people are dying these days of vitamin deficiencies, like scurvy, compared to the number dying from phytonutrient deficiencies? An estimated 7.8 million premature deaths are attributed annually to the inadequate consumption of fruits and vegetables, not getting at least 8 servings a day. Millions of lives hang in the balance being the scales that hang in the produce aisle. In the United States alone, if you add up all the fatal cancers, strokes, heart attacks, and other deaths that could have been averted by simply eating more fruits and vegetables, it comes out to about 450,000 deaths a year. There's a phytonutrient deficiency pandemic that could be wiped out with a few more daily servings of plants. Yet the pandemic is getting worse, not better. Over the last few decades, dietary quality has continued to deteriorate. Consumption of both fruits and vegetables, excluding potatoes, has dropped by more than half, and intake of legumes, also an important source of phytonutrients, has dropped by about 40%. At the same time, saturated fat consumption is on the rise. Only about 1 in 250 people even meet 80% of the American Heart Association's recommendations for a healthy diet. Perhaps people just don't understand the power of plants? Consider the first phytochemical, isolated in 1804 from the poppy plant, called morphine. In the 4th century, the first handbook of emergency medicine was published in China and recommended wormwood for malaria. 1,700 years later, the discovery was immortalized in a Nobel Prize in Medicine for the phytochemical artemisinin, and now included in the most effective combination therapies against the scourge of malaria. In my video c.nf/herbs to drugs, that's HERBS, the numeral 2, DRUGS, I dive into other powerful examples. Polyamory. Polyphenols are among the frontrunners in developing dietary approaches to fight age-associated diseases. More than 8,000 different polyphenols have been identified, but only a small proportion have had their health effects cataloged. Still, there's such a critical mass of data in favor of the protective benefits of these so-called lifespan essentials that recommended daily intakes of polyphenols have been proposed. I review what they can do and why in c.nf/polyphenols, where I also note the one source of flavonoids associated with an increased mortality, grapefruit, chalked up in part to grapefruit's suppression of a set of detoxification enzymes in our intestines. Natural geroprotectors. Geroprotectors are substances that increase longevity and or have other anti-aging properties. More than 200 have been found. See geroprotectors.org. Some of the most powerful ones that even beat out synthetic compounds are natural plant extracts of simple herbs and spices like celery seed. There are phytonutrients that can increase the maximum lifespan of animals by as much as 78%. Plant extracts that have been shown to increase the lifespan of lower organisms include acai, apples, including boring red delicious, asparagus, blueberry cinnamon, cocoa, corn, fenugreek seeds, grape skins, holy basil leaf, peach, pomegranate, rose, and turmeric. Fewer improve the lifespan of mammals, like mice, and those that do, like lemon, are tested on inbred strains chosen for their rapid aging. Many of the quote-unquote superfoods that can extend the lives of enfeebled mice have no significant effect on robust, long-living ones, and those that do may be a result of inadvertent dietary restriction. For example, mice fed a turmeric compound lived longer than control group mice, but they weighed about 3% less, suggesting they were eating less. Maybe they weren't curry fans. The dietary restriction alone could potentially account for the longevity. When researchers subsequently fed mice isocalorically instead of ad libitum, effectively forcing those groups of mice to eat the same amount, the turmeric benefit appeared to vanish. Speaking of dietary restriction-induced life extension, might the hormetic or xenohormetic benefits of phytonutrients undermine the longevity dividends of the stress of caloric restriction? A mixture of synthetic antioxidants completely blunted the life extension from a 20% dietary restriction in mice, but if you give polyphenols from blueberries, pomegranates, and green tea to intermittently fasted mice, they live even longer than had they just fasted intermittently. The longevity benefit was potentiated by phytonutrients. The researchers suggest that although alternate-day fasting in mice may have a net beneficial effect on lifespan, it also may entail harmful stressors that can be successfully countered by polyphenol intake. Plants, not pills. If phytonutrients can be so healthful, why not just take plant extract supplements rather than go to all the trouble of eating the plants themselves? Besides the misidentification, contamination, and adulteration issues rife within the poorly regulated supplement market we've discussed, there's a question of dose. taking polyphenol supplements can result in blood levels nearly an order of magnitude higher than that of a polyphenol-rich diet. When it comes to hormesis, less may be more. For a series of examples of how isolated phytochemicals in plant extracts can be life-extending at one dose but life-shortening at a higher one, check out c.nf/dosing. After all, many flavonoids function as nature's pesticide, protecting plants from predators like us. We co-evolved to counter these defenses, and thanks to hormesis, a smidgen of toxin can actually be beneficial, but a profusion of toxin can be toxic. To paraphrase a quote from a review on the anti-aging effects of polyphenols, it's easier to overdose on supplements than on salad. Synergy within plants. Some phytonutrients are so potent that functional doses can be encapsulated in a capsule, allowing for placebo-controlled trials of whole foods. For example, the sesame seed phytonutrient sesamin extends the lives of C. elegans and fruit flies. To see if it might have clinical effects, researchers pitted 2.5 grams a day of ground black sesame seeds, stuffed into capsules, against placebo. Within 1 month, less than a teaspoon of sesame seeds a day drove down systolic blood pressures by 8 points in middle-aged men and women. If sustained, that alone could decrease the risk of stroke by more than 25%. The downside of whole food studies is that you're never quite certain which component or components may be responsible. Was it the sesamin, or other sesame phytonutrients such as sesamol, sesamolin, or anthrasesamones A, B, C, D, E, and F? In a certain sense, who knows, who cares, right? As long as it works. It's hard to patent produce, which is perishable and relatively unprofitable, so big pharma and supplement companies, which are often one and the same, use a reductionist approach to try to uncover the quote-unquote magic bullet active ingredients of foods. But this ignores the concept of synergy. Sometimes the whole food is greater than the sum of its parts. For example, check out what happens when you pit various fractions of pomegranate polyphenols against prostate cancer cells in vitro. One subfraction reduced cancer cell growth by 30% compared to control, and another subfraction didn't help at all. The cancer grew as if it wasn't even there. So, mixing both together, you might expect the effect to land somewhere between the two, maybe 15% inhibition, with the ineffectual fraction washing out the better one? But no, put them together and you get 70% lower cancer growth. 30% plus 0% equals 70%. That's synergy, where 1 plus 1 is greater than 2. Pomegranate extract supplement that includes only one of the fractions would miss out on most or all of the benefit. When cranberry fractions were pitted against colon cancer cells, the discrepancy was even more extreme. Separately, 2 polyphenol fractions only suppressed cancer cell growth by 15% at most, but when they were combined into the total polyphenol complement of cranberries, colon cancer growth was suppressed by as much as 90%. Similar synergistic effects against human cancer cells in vitro have been found in components of ginger root, grape skins, rosemary leaf, and tomatoes. In c.nf/tomato synergy, I give a remarkable account of synergy in action. Basically, supplements of the red tomato pigment lycopene have repeatedly failed to successfully prevent or treat prostate cancer, but tomato sauce seemed to help. This makes sense, given studies showing how tomato components that are ineffective or worse individually can suddenly show anti-cancer effects when combined. For phytonutrients, plants are better than pills. To quote a past president of the American College of Lifestyle Medicine, quote, "The active ingredient in broccoli is broccoli," unquote. Synergy between plants. Each plant not only has thousands of different phytonutrients, but very different phytonutrient profiles, so there may be synergistic effects when eating different foods together, too. The reason it's better to get vitamin C in citrus form than in pill form is that you won't miss out on all those citrus phytonutrients like lemonin, lemonol, or tangerine that may interact, work together, and complement one another. But you'd also miss out on them if you instead ate an apple. Comparing apples and oranges is, well, like comparing apples and oranges. At least all fruits are fruits, whereas vegetables can be any other part of the plant. Roots harbor different phytonutrients than shoots. Carrots are roots, celery is a stem, dark green leafies are leaves, peas are pods, and cauliflower is true to its name, a collection of flower buds. Combining foods across different categories appears to increase the likelihood of synergy. The combined antioxidant power of raspberries and adzuki beans together, for example, is greater than just the sum of one plus the other. Neither soy phytonutrients nor green or black tea components alone decrease the tumor load or metastases of human prostate cancer implants in mice, but soy and tea together did. Hot pepper extracts alone appear to have little effect on the growth of cervical or breast cancer cells, but mixed with green tea, the cancer-killing power rose tenfold in the case of cervical cancer and 100-fold for breast cancer over green tea alone. These are interesting proof-of-principle studies, but they have limited human relevance if the cancer-stopping concentrations used in petri dishes couldn't be achieved in the bloodstream through normal dietary consumption. To resolve this issue, researchers exposed breast cancer cells from different patients to 6 different plant compounds individually, then altogether at the level you might find in your bloodstream after eating foods like broccoli, grapes, soybeans, and turmeric. While individual plant compounds had little or no effect on their own, in combination, dietary blood levels significantly suppressed breast cancer proliferation by more than 80%, inhibited cancer cell migration and invasion, stopped the cancer cells in their tracks, and eventually killed them all off. All the while, this so-called phytochemical super cocktail had no deleterious effects on the normal non-cancerous cells used as a control. A 10% tomato diet reduced the prostate cancer tumor burden of rats by 33%, and a 10% broccoli diet reduced the amount of tumors by 42%. Put them together, though, and a diet enhanced with both tomato and broccoli cut tumor levels by more than half. A spouse wrote to the editor of the Harvard Men's Health Watch, saying their husband, having heard about lycopene, wants to have pizza for his prostate, but they don't think it's a healthy food. The doctor replied with the suggestion of a, quote, cheese-free pizza with broccoli instead of pepperoni, please, unquote. Garden variety. Though there are generic plant compounds like vitamin C that are found scattered throughout the plant kingdom, there are also specific phytonutrients produced by specific plants to perform specific functions, both in their organs and in ours. We miss out on these if we're stuck in a fruit and vegetable rut, even if we're eating many servings a day. Airline pilots experience high rates of DNA damage due to being bombarded with radiation from the galaxy without the protection of the full atmosphere. A study found that pilots eating a greater mixture of phytonutrients had less chromosomal damage, but the researchers didn't control for total fruit and vegetable intake. Maybe the greater variety was just a proxy for greater quantity. Those randomized to eat 14 servings of fruits and vegetables a day for even just 2 weeks showed a reduction in oxidative DNA damage compared to those randomized to eat only 4 servings a day. But what about a study in which the number of servings is held constant and you just increase the diversity of the produce? That's exactly what a group of researchers in Colorado did. Both diets had the same number of daily servings, 8 to 10, but the high botanical diversity diet included fruits and vegetables from 18 different families, versus emphasizing just 5 in the low-diversity diet. Only those randomized to the high-diversity diet experienced a significant reduction in DNA damage. The researchers conclude that, quote, smaller amounts of many phytochemicals may have a greater potential to exert beneficial effects than larger amounts of fewer phytochemicals, unquote. Observational studies have also found that fruit and vegetable variety is associated with lower inflammation and better cognition, again, independent of quantity. Does this mixing and matching of a variety of plant foods actually translate into concrete difference for patients? Check out c.nf/food combining for a wild experiment involving secretly giving cancer patients a combination of a fruit, a vegetable, a spice, and a leaf, about 1/100th of a pomegranate, less than 1 floret of broccoli, less than 1/8 of a teaspoon of turmeric, and about 1/6 of a tea bag worth of green tea a day hidden in capsules and randomized against placebo. Surely such tiny amounts couldn't affect the progression of cancer, right? Wrong. As I show in the video, the cancer was significantly slowed down. Based on an update of the most extensive report on diet and cancer ever published, the foundation of cancer prevention is a diet centered around plants, whole grains, vegetables, fruits, and beans, while cutting down on alcohol, soda, meat, and processed junk. As I documented in How Not to Die, a completely plant-based diet may even shrink the tumor, not just slow down its growth, but there's no reason we can't do both with a plant-based diet chock full of particularly powerful plants. Moving from xenohormesis to microRNAs. If you thought the interspecies communication between the plant and animal kingdoms with xenohormesis was interesting, hold on to your hat. The central dogma of molecular biology has been challenged by a revolutionary 21st century discovery, microRNAs. Allow me to transport you back to high school biology. If you remember, our genetic code is stored in our DNA. Those are the instructions for creating and maintaining our body. There's no point in just having a set of blueprints if they can't be communicated to the builders to become manifest in the real world. RNA is that messenger. Messenger RNA transcribes a stretch of DNA code called a gene and has it translated into the finished product, a structural protein or an enzyme. The central dogma describes this flow of information as one gene to one messenger RNA to one protein. Then along came a shocking discovery from the Human Genome Project. Only about 2% of our DNA actually codes for proteins. So what does the other 98% do? When I was in medical school, the more than 1 billion letters of seemingly purposeless DNA were dismissed as noise or garbage sequences or junk DNA, perhaps just accumulated genetic schmutz from throughout our evolutionary past. That would seem a little wasteful, though. A parallel from astrophysics was drawn to this mystery, dark matter, the apparent fact that we can't account for about 85% of the matter in the universe. The mystery of the dark matter of our genome was solved in 2001. Most of our DNA violates the central dogma by being actively transcribed into non-coding RNA. That is, RNA that doesn't code for proteins. Then what does it do? We now know that there are more than 100 types of non-coding RNAs, but let's focus on the OG, microRNA. It takes a stretch of DNA thousands of letters long to encode the average messenger RNA. In contrast, microRNAs are only about 20 letters long. For example, the first microRNA ever discovered was 22 letters long in the 4-letter RNA alphabet: UUCCC, UGA, GAC, CUC, AAG, UG, UGA. What exactly do microRNAs do? They're generally created to glom onto messenger RNAs to prevent them from being translated into proteins. If DNA is the blueprint, and messenger RNAs are the construction workers translating those instructions into building parts of a house, microRNAs are like regulatory bureaucrats who intercede and keep particular workers from carrying out their duty. This is a good thing. Without building inspectors, minimum safety standards could be flouted, and different elements need to be timed properly. It makes sense to hold off on the roofers until well after the foundation is poured. Understanding microRNA regulation is particularly insightful because a single microRNA can block more than 1,000 different messenger RNAs. So one microRNA can effectively silence more than 1,000 different genes. In my building analogy, one simple instruction can put all the second-floor workers on hold until the first-floor workers finish construction. Then there are regulators who regulate the regulators, other non-coding RNAs that stop the microRNAs from stopping the messenger RNAs. But let's not even go there. Just when the complexity seemed overwhelming, researchers realized that even though there are 1 trillion possible microRNA 20 letters long combinations made from the 4-letter RNA alphabet, only a few thousand microRNAs seem to be active in the human body, and in any given cell, the 5 most abundant microRNAs average half the overall microRNA pool in a cell. However, in 2007, things got a lot more interesting. MicroRNAs have been found circulating in at least 12 different human bodily fluids. When I read that, I had to stop and think, wait, can I even name a dozen bodily fluids? We didn't think this possible since we have enzymes that chop up any floating RNA outside our cells as a caution against viruses, which often come bearing RNA. It turns out they're being transported in what are called exosomes, tiny bubble-like vessels that pinch off from cells. We used to think these budding blebs were just a waste disposal device for cells. Why, scientists seem to just jump to junk when they don't understand something. But in 2007, we discovered that they were packed with microRNA. Our cells were communicating with each other. In this way, a liver cell could send out microRNAs to regulate the genes in a lung cell, which could then regulate a brain cell, or vice versa. They could even talk to the next generation by depositing their microRNA load into a sperm or egg cell. What's the upshot of all this? It's now safe to say that microRNAs probably regulate virtually every biological process, playing essential roles in virtually every aspect of health. Mice genetically engineered to be unable to make microRNAs never even make it past the embryo stage. Diseases of all shapes and sizes have been linked to the dysregulation of microRNAs. But the good news is that there's something we can do about it. MicroRNA expression can be modified through diet. MicroRNAs and aging. What does this have to do with aging? As a major regulator of all cellular pathways, it would make sense for microRNAs to play a role, but the connection has special salience. The very first microRNA was discovered in the humble roundworm C. elegans. Guess what it did? Regulated its lifespan. Reducing the activity of the simple microRNA reduces lifespan and accelerates tissue aging, whereas overexpressing it significantly extends life. It turns out that the target of the microRNA was a DAF16 suppressor gene, and DAF16 is the worm equivalent of the FOXO gene, which can confer immortality to certain primitive animals, and is one of the most important genetic determinants of extreme longevity in humans. By blocking the repression of this longevity gene, the microRNA had a lifespan extending effect. Knowing the expression patterns of just a few microRNAs in C. elegans can effectively predict the longevity of individual animals. To study the effects of microRNAs on mammalian lifespan, a series of lifestyle interventions were set up in mice. One group of mice was put on a high-fat diet, and they lived 101 weeks. A second group was put on a high-fat diet with added voluntary exercise, and they lived for 114 weeks. The next group was put on a low-fat diet, which brought them up to 127 weeks. Fourth group was put on a low-fat diet plus exercise, lived 131 weeks. Group 5 was put on a caloric restriction on a high-fat diet, lived 137 weeks. And finally, mice were put on caloric restriction on a low-fat diet, lived 153 weeks, a lifespan more than 50% longer than those in the regular high-fat group. Using this approach, the researchers found that 92 microRNAs were correlated with lifespan, including 84 in an inverse manner. In other words, the microRNAs generally appeared to be suppressing longevity genes, so certain microRNA levels were up to 90% lower in the longest-living group. However, there are exceptions. For example, microRNA 17 directly extends the lifespan of mice. Transgenic mice created to overexpress the microRNA live longer and healthier lives, proving that microRNA isn't just correlated with a longer life, but directly causes it, in part by repressing mTOR, which I talked about earlier. Such so-called longevity microRNAs may account for the parabiosis findings. Remember the mad scientist experiments of rejuvenating old animals by sewing them to younger cage mates and hooking together their circulations? That effectively proved that there are blood-borne determinants of aging. Perhaps microRNAs fit the bill. In humans, dozens of circulating microRNAs are upregulated as we age, and dozens are downregulated. The blood levels of 7 microRNAs may be able to differentiate Alzheimer's patients from healthy controls with up to 95% accuracy. If these dynamics are all just genetic, then microRNA levels could still be useful as biomarkers or diagnostics, but they might be harder to tweak to control our fate. But no, a study on identical twins who died about a decade apart found that they had highly discordant microRNA levels, suggesting that non-genetic factors such as diet and lifestyle are pivotal in microRNAs related to life expectancy. Exercise power over microRNAs? More than 6,000 patents have been filed on the potential use of synthetic microRNA mimics and inhibitors to fight aging and disease, but to date no such drugs have been approved. Is there anything we can do naturally? MicroRNAs may be one of the reasons randomized controlled trials have shown that exercise can prevent cognitive decline in older adults and improve cognition for those already suffering from Alzheimer's disease. There are microRNAs that are reduced in Alzheimer's, like microRNA 132 and microRNA 338, but boosted by exercise. And conversely, there are microRNAs that are overexpressed in Alzheimer's, like number 7 and 766, but reduced by exercise. The picture isn't completely clear, though. MicroRNA 146A has consistently been found to be elevated in the blood, brain, and cerebrospinal fluid of Alzheimer's patients, and although acute resistance training and chronic basketball training were found to reduce circulating levels of microRNA, rowing training and marathon running were found to increase them. So we still have much to tease out about the potential role that microRNAs play in the way physical activity can improve mental activity. Modulating microRNAs with meals. MicroRNAs may also be a mediator of the benefits of polyphenols. A dozen different phytonutrients have been shown to change the expression of dozens of microRNAs in vitro. As we know, one problem with Petri dish studies is that sometimes concentrations are used that far exceed that which can be achieved through regular dietary consumption, but a few foods have been put to the test. For example, a study showing that extra virgin olive oil with high polyphenol content has a different microRNA impact than lower polyphenol olive oil suggests that the polyphenols may be playing an active role. Nuts, either 1 to 2 handfuls of walnuts a day for a year, or a single handful of a combination of almonds and walnuts for 8 weeks, also change the levels of an array of microRNAs in the bloodstream. But to what end? There are well-known inflammatory microRNAs, like microRNA 155, that are suppressed by a variety of flavonoids, genistein in soy, quercetin in apples and onions, allylisocyanate in the onion family of vegetables, curcumin in turmeric, and apigenin in parsley, celery, and chamomile tea. MicroRNA 155 also plays a role in cancer. For example, it's implicated in the development of acute myeloid lymphoma, the deadliest form of leukemia, and the most common acute leukemia among adults. In a study entitled Alleviating the Progression of Acute Myeloid Leukemia by Sulforaphane Through Controlling MicroRNA 155 Levels, not only could microRNA 155 levels be dropped by about 80% by the cruciferous vegetable compound in vitro, but this led to a significant drop in cancer cell viability. Unfortunately, broccoli sprouts, the most concentrated source of sulforaphane, have yet to be tested on AML patients to assess clinical outcomes. Flavonoids have been found to suppress the proliferation of tumor cells by both suppressing oncogenic or cancer-causing microRNAs and boosting tumor suppressor microRNAs. Long-term soy consumption in breast cancer patients had this effect, perhaps helping to explain why soy consumption appears to help prevent the development of breast cancer in pre- and postmenopausal women, as well as improve survival in breast cancer patients and reduce the chances of the cancer coming back. This may also explain the upregulation of tumor suppression microRNAs in vegetarians and vegans compared to omnivores, and the subsequent lower cancer risk, though meat consumption can affect microRNAs too. Rectal biopsies, taken before and after a month of eating 3 daily servings of beef or lamb, found significant upregulation of oncogenic microRNA clusters in rectal tissue. Adding resistant starch to the diet was able to reduce, but not completely eliminate, this effect. Similarly, the cooked meat carcinogen PHIP, found particularly in grilled, broiled, fried, and barbecued chicken, causes estrogen-like effects on microRNAs, implicating the initiation and progression of breast cancer. MicroRNA modulation has also been used to explain why saturated fat increases insulin resistance, though so far this has only been demonstrated in rat muscle. Beyond potentially contributing to the lower cancer and diabetes rates among those eating plant-based, diet-induced microRNA changes may also contribute directly to life extension. A study on circulating microRNA expression in the Loma Linda Blue Zone, where healthy Adventist vegetarians live about a decade longer than their fellow Californians, found half a dozen microRNAs related to aging differentially expressed between vegetarians and non-vegetarians that potentially provide mechanisms for the longer plant-based life expectancy. Interestingly, for one of the anti-aging measures, semi-vegetarians and vegans both beat out ovo-lacto-vegetarians, those who eschew meat but eat eggs and dairy. Semi-vegetarians were defined as eating meat at least once a month, but no more than once a week. The researchers suggest that they might have ended up eating fewer animal products overall than the vegetarians who more regularly ate eggs and dairy. XenomicroRNAs. Intercellular microRNA communication is conserved throughout the evolutionary tree of life, raising the possibility of cross-kingdom gene regulation. In the 18th century, life was classified as belonging to either the plant kingdom or the animal kingdom. In the 19th century, single-celled organisms like amoebas got their own kingdom, and with further improvements in microscopy, bacteria got one as well. These days we're up to 7 kingdoms. Algae and fungi each got their own, as did bacteria-like organisms originally described as extremophiles, living in previously thought uninhabitable zones like hot springs. With a common language of microRNAs, might inhabitants of different kingdoms communicate with one another? In 2011, we learned that microbiome microRNAs could modulate the gene expression of their host. For example, there are gum disease bacteria that secrete vesicles filled with microRNAs shown to penetrate host cells, apparently to suppress our immune response. Sneaky! Then in 2016, we learned that we have our own microRNA counterinsurgency program. Fecal microRNAs produced by our intestinal lining cells infiltrate our gut bacteria, regulating their gene expression and growth, and may be essential for the maintenance of a healthy microbiome. If there's microRNA manipulation happening between the simplest and most complex organisms on Earth, what about crosstalk with an intermediate, the plant kingdom? The Nerds cartoonist Randall Monroe at xkcd.com used a comic captioned, "Really, every gathering is a family reunion," to remind us that ultimately we're all related. If you go back far enough, each of us can trace back to a common ancestor, all the way back to the very first Homo sapiens to which we're all related. So the cartoon shows a party scene with stick figures labeled me, second cousin, 14th cousin, and 35th cousin, and also a pet cat labeled 17 millionth cousin. Yes, if you go back far enough, you and Fluffy actually had a real flesh and blood common ancestor. The cartoon also has a houseplant labeled 50 billionth cousin. Using molecular clock dating techniques on shared DNA deviation, it's estimated that plants and animals diverged 1.576 billion years ago, give or take 88 million years. So even you, Fluffy, and the ficus share an ancestor. Family reunion indeed. Recognition of the ubiquitous presence and activity of microRNA in plants followed soon after their discovery in animals. Cotton plants, for example, use microRNAs to silence the virulence genes of a pathogenic fungus. What influence might plant microRNAs be having in another cross-kingdom interaction with us? Just like we share many microRNAs with other animals, some microRNA sequences in plants have such close overlap with animal microRNAs that scientists suspect they're actually the same microRNA conserved through 1.5 billion years of evolution. Regardless, matching up the sequences of plant microRNA to human messenger RNA, there appear to be at least 1,000 different human genes that plant microRNAs could target. Plant-based diets contain thousands of biologically active microRNAs. While the scientific community has historically chalked up the benefits of fruits, vegetables, and herbal medicines to the presence of phytonutrients, it may be their microRNAs playing a role. Isolated phytonutrients have often failed to fully replicate the effects of whole foods from which they were extracted. This failure has been attributed to the synergistic symphony of interactions between the various components working together. As we've seen, one way that phytonutrients like polyphenols affect our physiology is by manipulating our microRNA expression, but perhaps plant microRNAs are silencing our genes directly. The exploration of the potential for cross-kingdom gene regulation with so-called xenomicroRNAs from plants is currently considered among the most exciting topics in all of science. Broadly, the concept of cross-kingdom genetic manipulation is nothing new. After all, RNA and DNA from viruses have been hijacking human cells since time immemorial. But if food-derived microRNAs are altering our gene expression, that certainly offers new meaning to the phrase, "you are what you eat." Dietary microRNAs. Dietary microRNAs could mean that food may provide information as well as nutrition, information that could effectively switch our genes on or off. Some researchers have conceptualized dietary microRNAs as so-called dark nutrients, in another nod to dark matter, claiming that they may play a significant role in human health. Yes, plant microRNAs have been shown to enter human cells and alter our gene expression, but let's take a step back. Would microRNAs in our diet even survive cooking or digestion? Some processed plant products, such as olive oil and beer, seem to have lost their microRNAs in the production process. What about microRNA loss on the stove? We used to think cooking destroyed genetic material, but more recent experiments show that some plant microRNAs can take the heat. Some, such as the microRNA 159 in broccoli, remain stable after cooking, while those like artichoke 319 are partially destroyed. And the levels of other microRNAs, like those found in cooked beans and brown rice, rise even higher after cooking, presumably by being liberated into the cooking water. Mammalian and avian microRNAs in meat, dairy, and eggs survive cooking and processing, based on studies of pork and poultry sausages, ham, salami, hard-boiled eggs, cheese, and pasteurized milk. There's little change in microRNA levels between raw beef and beef roasted until well done. However, they still would have to survive the acid bath of the stomach. Again, the conventional wisdom was that microRNAs would be destroyed during digestion, but if you dunk them in acidic gastric fluid, the majority of plant and animal microRNAs appear to survive for at least 6 hours. In the small intestine, though, there are RNases, enzymes that chew up naked RNA. How might they survive that gauntlet? They may not have to. A study in mice found that the stomach itself appears to be the primary site of dietary microRNA absorption into the bloodstream. Alternately, microRNA may travel wrapped in protective exosomes. From plants, exosome-like vesicles have become known as quote-unquote edible nanoparticles, and they can be filled with microRNAs. A pound of fruit can contain 1 gram of these little delivery vehicles. This packaging has been proposed as a solution to the microRNA bioavailability problem. In that form, they could be absorbed by our intestinal lining, repackaged into exosomes, then released into our circulation. The proof is in the pudding, though. When we eat microRNAs, do they show up in our bloodstream? Unlike typical animal microRNAs, the tips of plant microRNAs are tagged with a methyl group. This not only makes them more resistant to digestion, but it allows researchers to differentiate them from pre-existing microRNAs circulating in animals. Feed mice some cruciferous vegetables, and cruciferous vegetable microRNAs peak in their bloodstream within 6 hours and can be picked up in multiple organs. Corn microRNAs peak in the bloodstream of pigs fed fresh corn between 6 and 12 hours after consumption. Most plant microRNAs are carried in exosomes, which can even transport RNA into the brain. The cruciferous microRNAs were found circulating for more than 36 hours. What about in people? Researchers found that as many as 5% of all detectable microRNAs circulating in people's bodies may be from plants. The first report of plant microRNAs circulating in humans was published in 2012. The consistent finding of rice microRNAs in the bloodstreams of Chinese consumers. Just as fish-eating seals have circulating fish microRNAs and cows have plant microRNAs from foraged crops and grasses, most of the plant microRNAs in us are from fruit and vegetable species. Plant microRNAs have been found throughout the human body, including the brain, breasts, kidney, liver, and lungs, as well as in breast milk, amniotic fluid, and umbilical cord blood. Are these just incidental findings, or are dietary microRNAs doing anything to us or for us? Fruit and vegetable microRNAs. Hundreds of different microRNAs have been found in the edible nanoparticles of common fruits and vegetables. In a proof-of-principle study, edible nanoparticles from grapes were fed to mice. They were taken up in intestinal cells, changed gene expression, and protected the mice from gut inflammation. Similar experiments with carrot, ginger, and grapefruit nanoparticles found a range of beneficial regulatory effects. But how do we know it was necessarily the microRNAs? MicroRNAs are such simple molecules that we can make them from scratch. So researchers synthetically made strawberry microRNA 156, rice microRNA 168, and cabbage microRNA 874 to isolate out microRNA-specific effects. And indeed, they were found to have anti-inflammatory effects in human cells. RNA extracts of blueberries, raspberries, and apple peels had a similar effect. To make sure this was not a generic RNA effect, an RNA extract from beef beef was tested, and it failed to dampen inflammation. One plant microRNA found circulating in people is microRNA 156A. Decreased levels were found in the blood and blood vessels of patients with cardiovascular disease, suggesting it might have a protective effect. But where is microRNA 156A concentrated? In green vegetables. Give people a salad and you can see a bump in 156A within an hour. Might lower levels of 156A in heart disease patients just be a proxy for low levels of greens intake? To find out if it was actually cause and effect, researchers exposed human artery endothelial cells to pure, synthesized microRNA 156A and showed that it targets a sticky protein called junctional adhesion molecule A that abets in the attraction of inflammatory immune cells into the artery wall to trigger atherosclerotic plaques. And indeed, boosting microRNA 156A reduced the attachment of inflammatory cells to artery lining walls. So the protective effects of green vegetables on cardiovascular disease may be more than just a nitrate effect. A similar story was found with breast cancer and microRNA 159A, which is found in abundance in broccoli. Lower blood levels correlated with higher breast cancer incidence and tumor progression. MicroRNA 159A wasn't just a broccoli biomarker, though, but an active player targeting a cancer-promoting gene called transcription factor 7. When mice implanted with human breast tumors were fed straight microRNA 159A, they experienced a dramatic decrease in tumor weight and growth. So the protective effects of cruciferous vegetables on breast cancer may be more than just a sulforaphane effect. Herbal microRNAs. Might some of the effects of herbal medicines also be ascribed to plant microRNAs? In c.nf/herbal miRNAs, I review the evidence on microRNAs in ginseng, licorice, red sage, or danchen, and another traditional Chinese herb, honeysuckle, which appeared to have remarkable efficacy in hospitalized COVID patients. Unfortunately, as I explained in the video, like so many impromptu pandemic trials, the study left a lot to be desired. The bottom line is that microRNAs may shed some light on how plants can be so powerful, including why some poisonous plants are so poisonous. Apple of discord. The concept that dietary microRNAs could be therapeutic has been called compelling, fresh, and revolutionary. However, the first reports were met with suitable skepticism, which have since evolved into a fierce controversy. Many subsequent attempts at replication failed to unambiguously confirm the initial findings, leaving the medical literature littered with editorials with titles like Diet-Derived MicroRNAs Unicorn or Silver Bullet, and Dietary Non-Coding RNAs from Plants Fairy Tale or Treasure. I run through the tug-of-war controversy in c.nf/discord. Though it continues to be an exciting area, the biological role of dietary plant microRNAs remains far from being firmly established. Eating animal microRNAs. What about eating or drinking animal microRNAs present in meat, milk, and eggs? Animal-derived microRNAs can sometimes be absorbed in much more significant amounts than plant microRNAs. The problem is it's much more difficult experimentally to distinguish between dietary animal microRNAs and the ones our own animal body makes, since they can be nearly or completely identical. One way researchers have tried to solve this conundrum is to genetically engineer so-called knockout mice, in which the gene for a particular microRNA is knocked out, inactivated, or deleted. For example, researchers made microRNA 451 knockout mice drink the blood of wild mice, chicken, and pigs. When they then found microRNA 451 circulating in their bloodstreams and carrying out its regulatory function, they knew that ingested animal microRNAs could indeed affect physiology. Vampiric mice aside, how could confirmatory experiments be carried out in people? This is an important question, since there are a number of pro-inflammaging and cancer-promoting microRNAs in animal products that are 100% matched with the same microRNAs in humans. Even if you couldn't differentiate meat microRNAs from me microRNAs, you could at least test to see if you get a rise in blood levels after consumption. Tracking 3 shared cow microRNAs in people after eating beef failed to show a bloodstream bump, though if you remember that rectal biopsy study did at least show microRNA changes down in the colon after red meat consumption. Chicken microRNAs from egg consumption, however, can be detected in the human bloodstream after intake. In the USDA-funded study published as "MicroRNAs in Chicken Eggs are bioavailable in healthy adults and can modulate mRNA expression in peripheral blood mononuclear cells," volunteers were fed hard-boiled eggs. Within 9 hours, blood levels of microRNAs 181A and 181B rose to about 150% and 300% above baseline respectively, reflecting their relative abundance in eggs. This was accompanied by a suppression of the validated gene target of 181B in their white blood cells. To verify that chicken microRNAs actually made it into the human bloodstream after egg consumption and don't just indirectly bump up endogenous microRNA levels, the researchers were able to track the entry of a chicken-specific microRNA into their circulation. Drinking animal microRNAs. The greatest evidence for the potential for cross-kingdom gene regulation comes from the dairy literature. Of all body fluids tested, milk contains the largest load of microRNAs. Milk is a secretory product of mammary gland epithelial cells which discharge microRNA-packed exosomes into the milk. Based on the human breast milk literature, most are immune modulatory, especially during the first 6 months of lactation. We've long known that breast milk contains antibodies and other protective agents missing from baby formula to provide passive immunity and help with immune system development, but microRNAs may add additional urgency to the entreaty that breast is best. Babies aren't just breastfed, but breast-programmed. Milk is no longer perceived as just a food for infants, but rather a highly sophisticated communication system orchestrating early development. For example, we've known for more than a decade that something in milk prevents allergies. Rat milk, but not rat formula, prevents allergies in rat pups. MicroRNAs may help explain why breastfeeding seems to protect against childhood asthma and pediatric infections and results in higher intelligence compared to formula feeding. If milk microRNAs can so manipulate our infant's physiology, what happens if we drink milk after weaning as an adult, or even drink milk from another species? The milk of pandas and pigs, humans and cows and water buffalo share a few common highly expressed microRNAs, but cow's milk also contains large quantities of hundreds more, as many as 1,500 different microRNAs. Because most milk microRNAs are encapsulated in exosomes, they're resistant to heat, while most exosomes and their cargo are destroyed by boiling or ultra-high temperature processing used to make shelf-stable creamers. Commercial pasteurization leaves a significant proportion of milk microRNAs intact. Most then survive conditions of digestion in adults. To prove that milk microRNAs from one species can make it into the circulation of another species who drinks it, milk microRNAs were tagged with a fluorescent label as a tracker. Loaded into cow's milk, the microRNAs ended up distributing and accumulating in the spleens, livers, hearts, and brains of mice. In vitro, human cells take them up and have multiple genes upregulated and downregulated as a result. Of course, mice suckling from cows is ludicrous. Primates, on the other hand, made for each udder. Government-funded researchers at the University of Nebraska had men and women drink different quantities of dairy milk, 1, 2, or 4 cups. Considerable amounts of milk microRNAs appeared in their blood in a dose-dependent manner, peaking within 4 hours after consumption and affecting target gene expression. The bovine microRNAs tested were identical to human microRNAs, though. How do we know that drinking milk doesn't somehow boost the endogenous production of our own microRNAs instead of crossing over from the digestive tract into our bloodstream? The levels of a control microRNA not found in milk were unaffected, but more robust evidence came from subsequent studies using highly sensitive PCR techniques that could detect tiny differences between bovine and human microRNAs. And indeed, blood concentrations of bovine-specific microRNAs end up circulating throughout our body within hours of milk consumption, providing compelling evidence that dairy milk exosomes from commercial pasteurized milk right off store shelves can end up in the tissues of human consumers. What consequences may this have? The most abundant microRNA in milk is microRNA 148A, and it's a key inhibitor of crucial suppressors to the engine of aging enzyme mTOR that I talked about before. After all, what does an infant need more than accelerated aging? This may be even more apparent in dairy cows, whose newborns double their birth weight in 40 days, more than 4 times faster than our infants. Cows have been selectively bred for lactation performance, which incidentally appears to have exaggerated microRNA 148A expression. The species-specific growth stimulation programmed by milk microRNAs was meant to be confined to infancy. The concern is that continuous exposure to growth-promoting exosomes of pasteurized milk may confer substantial risk for the development of chronic diseases, from acne and obesity to diabetes and cancer. MicroRNA 148A, for instance, directly stimulates prostate cancer cell growth in vitro, which may help explain why dripping milk on human prostate cancer cells boosts their growth rate by more than 30%. Maybe that's why a systemic review of observational studies report that an overwhelming majority of them, 19 out of 20, found a link between milk consumption and increased risk of developing prostate cancer. MicroRNA 21, one of the earliest identified cancer-promoting oncomicroRNAs, is also a signature microRNA of dairy milk. MicroRNAs may also help explain the difference in associated mortality between fresh milk and fermented milk in 2 large Swedish studies. Significant increases in mortality risk in men and women were associated with the consumption of fresh milk but not soured milk. Bacterial fermentation of milk can lead to exosome and microRNA breakdown, though this does not appear to affect prostate cancer risk, which seem to be elevated by both milk and yogurt consumption. A recent review titled "Cow's Milk May Be Delivering Potentially Harmful Undetected Cargoes to Humans" suggested that dairy recommendations need to be reconsidered in light of the fact that an estimated 35 trillion bovine exosomes are floating around in each glass of milk. Given the role of exosomes in pasteurized milk to boost mTOR activity, some researchers have concluded that, quote, milk exosomes should not reach the human food chain, unquote, as milk, quote, unquote, is not a suitable food for adults. In other words, milk is for babies.